DNA vaccination. A DNA vaccine is a genetic vaccine that uses a small circular DNA molecule, plasmid, containing the gene for a specific antigen, a protein from a virus, bacterium, or parasite. When this DNA is injected into the host's own cells produce the antigen, which stimulates the immune system, mimicking a natural infection but without causing disease. Process of designing a DNA vaccine. Designing a DNA vaccine involves several key stages. 1. Antigen selection. The first step is to identify a protective antigen gene from the pathogen, virus, bacteria, or parasite. This gene encodes a protein that can trigger a strong immune response, such as a surface or envelope protein. 2. Gene cloning. The selected gene is inserted, cloned, into a plasmid vector, a small, circular DNA molecule used to deliver genetic information. The plasmid includes a strong eukaryotic promoter, example CMV promoter, for transcription in host cells. A polyadenylation signal for mRNA stability. An antibiotic resistance gene for bacterial selection. 3. Plasmid amplification and purification. The recombinant plasmid is multiplied in E. coli bacteria and then purified under sterile conditions to be used as a vaccine. 4. Vaccine delivery. The purified plasmid DNA is delivered into the body using intramuscular injection, intradermal injection, gene gun, biolistic delivery. Electroporation. Short electrical pulses increase cell membrane permeability, enhancing DNA uptake. 5. Expression of antigen in host cells. Inside the body. The plasmid DNA enters host cells, mainly muscle or dendritic cells. It moves into the nucleus but does not integrate into host DNA. The inserted gene is transcribed into mRNA. The mRNA travels to the cytoplasm and is translated into the antigenic protein. 6. Immune system activation. The antigen is processed and presented to immune cells via MHC class I molecules. Rightward arrow activates cytotoxic T cells, CD8. MHC class II molecules. Rightward arrow activates helper T cells, CD4, and B cells. This leads to both antibody production and T-cell mediated immunity. Immune response after DNA vaccination. DNA vaccines activate both arms of the immune system. 1. Cellular immune response. Antigens synthesized inside the host cell are presented via MHC-I to CD8 cytotoxic T cells. These cells destroy infected or antigen-expressing cells and generate memory T cells. 2. Humoral immune response. Antigens secreted or displayed on the surface of cells are recognized by B cells. With help from CD4 helper T cells, MHC2, B cells differentiate into plasma cells that produce specific antibodies to neutralize the pathogen. 3. Memory response. The vaccine induces long-term immunological memory, ensuring a faster and stronger response upon future exposure to the real pathogen. Fate of the plasmid in the host cell. 1. Entry and expression. After injection, plasmid DNA enters some host cells, travels to the nucleus, and directs antigen production. 2. No integration into host genome. The plasmid DNA remains episomal, free in the nucleus, and does not integrate into the host's chromosomes. This ensures no genetic modification of the host genome. 3. Temporary existence. The plasmid does not replicate along with host DNA. Over time, it is naturally degraded by cellular enzymes. As the transfected cells die naturally, the plasmid is also eliminated from the body. DNA plasmid does not remain forever in the host, it stays only long enough to produce antigens and trigger immune responses. Advantages of DNA vaccines. Safe, no live or attenuated pathogens used. Stable, DNA is stable at room temperature, no strict cold chain needed. Easy and fast to produce, simple bacterial production and rapid design for new diseases. Broad immunity, induces both humoral and cell-mediated immune responses. Long-lasting protection, generates memory T cells and B cells. No need for adjuvants, plasmid DNA itself acts as an adjuvant due to CpG motifs. Flexible design, multiple antigens can be encoded in a single plasmid. Limitations of DNA vaccines. Low uptake efficiency, DNA uptake by human cells is often poor. Weak immune response in humans, highly effective in animals, but less potent in humans, still improving. Need for delivery aids, often requires electroporation or gene gun for efficiency. Possible integration risk, very rare but theoretical chance of plasmid integration into host genome. Autoimmunity concern, rare, anti-DNA antibodies could theoretically develop. Multiple doses may be required, to achieve sufficient immunity. Examples of DNA vaccines. Disease. Target antigen. Vaccine name/developer. Status. COVID-19. Spike, S, protein of SARS-CoV-2. ZyCoV-D, Zydus, India. Approved. Zika virus. Envelope protein. GLS 5700, Inovio Pharmaceuticals. Clinical trials. Influenza. Hemagglutinin, HA. Various DNA constructs. Experimental. HPV, human papillomavirus. L1/L2 capsid proteins. Research stage. Experimental.